In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum. How are you, dear students? I welcome you in Pakistan International School's virtual learning class for first year biology. Dear students, today we will discuss carbohydrates and its second type, oligosaccharides. We have discussed monosaccharides in the previous lecture, which is on chapter number 2, biological molecules. So let's get started. Dear students, if you see brief summary and classification nomenclature of carbohydrates, so we can see that there are three kinds of carbohydrates: monosaccharides, oligosaccharides, and polysaccharides. So monosaccharides are having two functional groups. Some are having aldose group and some are having keto group. So the example of aldose group is glucose and ketose group is fructose, which we have discussed earlier. If we talk about number of carbon atoms, so monosaccharides are having the least one triose, three carbon atoms. So they are trioses, then tetroses, pentoses, and hexoses and heptoses. If we talk about oligosaccharides, it is having uh, like two, if they are having two monosaccharides units, that's why they are called disaccharides. These are the most commonly found disaccharides. Then if they are having three monosaccharides units, trisaccharides. If they are having four saccharides unit, we are having tetrasaccharides, and so on. So we can have the examples of disaccharides: maltose, lactose, and sucrose. uh, Trisaccharides is raffinose, and tetrasaccharides are stachyose. If we talk about polysaccharides, so they are of two types: homopolysaccharides and heteropolysaccharides. Homo means same. When all the units, all the monosaccharide units of polysaccharides are same, for example glucose, 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 so they are called monosaccharides. And in that we are having the example starch, dextrin, glycogen, cellulose, and inulin. And heteropolysaccharides means they are having different units of monosaccharides in that. So we are having the examples: hyaluronic acid, heparin, chondroitin sulfate, dermatan sulfate, and keratin sulfate. Now we will see oligosaccharides in detail. If we just see oligosaccharides, so we can see these groups consist of derivatives of monosaccharides. It means when monosaccharides are combined together, so they will form oligosaccharides. The least one is two units of monosaccharides. When they unite together, they are called disaccharide, according to the number of the monosaccharide units which are present in them. So the highest in oligosaccharides, 10 monosaccharide units can be united one with one another upon condensation. Upon hydrolysis, it can give 2 to 10 polysaccharides, monosaccharide units. So the most common are disaccharides. So disaccharides means di means two. So saccharides means carbohydrates. So when two carbohydrates or two monosaccharide units come closer by condensation or removal of water, so what will happen? Disaccharides are formed. If we talk about their properties, they are less sweet in taste and they are less soluble in water if we compare with monosaccharides, and they can be hydrolyzed into monosaccharides. Definitely when they broken down, the bonds are broken down, so um, uh, they will form monosaccharides. The most common examples are maltose, lactose, and sucrose, and its general formula is C12H22O11. The very common example is sucrose because this is mostly present in the plants, and it is called transport disaccharides because when in the plants by photosynthesis, what is formed? Glucose is formed. We know glucose is formed. So glucose is transformed into, or by condensation it forms like sucrose. Okay, it is transport from the leaves to other parts, like to the roots, to the fruits, vegetables, so in the form of sucrose. Then it can, like sucrose is present in considerable amount in sugar beet, pineapple, sorghum, and all sweet fruits. So they are widely used as sweeteners at home for making sweet dishes as well. So sucrose is actually mostly abundantly found in the plants. So what happened on hydrolysis, it gives two different types of monosaccharides. One is alpha glucose and other is beta fructose. You know that what is alpha glucose and beta glucose, we have discussed in the previous lecture. We will just see in a few minutes. So what happened here? In the diagram, you can see formation of sucrose because two monosaccharides, glucose and fructose, are there. So alpha glucose and beta fructose. It means alpha means when hydroxyl group is present on the first carbon, but it is a giant below, at the below orientation. This is called alpha glucose. And in fructose, when like this hydroxyl group is present up in the orientation on carbon number 2. Carbon number 1 is present here outside of the here carbon number 2 is 1 is present, and with carbon number 1 not, but actually carbon number 2 we are having what? Hydroxyl groups. So and it is above in the orientation, that's why it is called beta fructose. Fructose are the circular rings. Fructose is a circular ring which is having 6 carbons, but 5 are in like 5 are in the, what we can say, in the ring. Sixth one is outside, but it is all connected with O group and it forms 5 corner ring, and glucose is a 6 corner ring. Okay, so when this one, hydrogen from fructose and hydroxyl group from glucose, by condensation, they combine and they form water molecule, which is removed out, and there is the bond that is called glycosidic bond. Now glycosidic linkage or glycosidic bond means the bonds between two carbohydrate molecules, two monosaccharides. So this is alpha 1 2. It means alpha means the first carbon group of glucose is having hydroxyl group on below in the orientation. That's why it's called alpha first. Okay, so alpha 1 and then 2 is here. Okay, on which it is 2 here, and then they form alpha 1 2 glycosidic linkage. Next example is maltose. If we talk about maltose, so we can see here it is commonly found in malt sugar. Means in most of the grains it is present, and by it is, and also the very important property that it is an intermediate disaccharide produced during the breakdown of starches and glycogen. Starch is Starch is the stored food in the plants, and glycogen is the stored food in animals. So when they break down the intermediate compound, that is called what? Maltose, and it is also found in germinating seeds and lots of grains, that is in the um, what we can say, barley, yes, and the barley and barley starch that is used for in the brewing industry for the production of alcohol, and it is as well as used for the malting process. This is because maltose is used in this process, so that's why it is called malting. And by the enzyme amylase, because we know that carbohydrate is broken down by the enzyme amylase. It yields two molecules of alpha glucose. Alpha glucose, as I have told you before, that in which hydroxyl group is present below the in orientation at carbon number 1, and on the other at carbon number 4. So the carbon number 4 and carbon number 1, alpha glucose, and this is alpha glucose also, because here mm, here at carbon number 1 hydroxyl group is present below in orientation, here also at 1 below orientation, but they are linked with carbon number 1 and 4. The hydroxyl group from here, it is joined with hydrogen, and hydroxyl group are joined like this, and they form water in the process of condensation, and they form what linkage? 1,4 glycosidic linkage. Okay, so let's talk about lactose. It is commonly found as milk sugar in the milk of mammals. So 4 to 6% in cow's milk and 5 to 8% in human milk. And it is also the byproduct to manufacture cheese. Yes, we know that cheese is also produced in the dairy by milk in the dairy industries. So it yields on hydrolysis into beta galactose and a molecule alpha glucose. So how it is done, we will see here. You can see this one, that this is galactose, okay, and this is beta glucose. So in that, this hydroxyl group and above the orientation, that's why it is called beta, and here it is also above the in orientation, so that's why it is called beta glucose. So both are beta glucose. So actually here it is some mistake in the book, that is beta glucose. So they also form 1 and 4 linkage, glycosidic linkage, and water is removed out. So on condensation, it gives what? It is broken down into two molecules of monosaccharides. These carbohydrates are also used as reducing sugar and non-reducing sugars. So because the carbohydrate, which is capable of being oxidized, like itself it is oxidized, means oxygen is added or electrons are added, and causes the reduction of other molecules without having to be hydrolyzed first. They don't hydrolyze, but they absorb electrons from the other compound. They reduce other substances and get oxidized by itself. Those are called reducing sugars. and opposite of this, which are unable to be oxidized and do not reduce other substances, these are non-reducing sugars. So all monosaccharides and 2 of 3 disaccharides like maltose and lactose, they are like they are reducing sugars due to its chemical structure, and they are like used in some metabolic reactions in the body as well. Third type of disaccharide like sucrose and polysaccharides are non-reducing agents. So dear students, that's it for oligosaccharides. I hope you have understood this, the linkage and how they are alpha and beta and how they are hydrolyzed and condensation is done. So inshallah, we will see you in the next lecture and continue the next topic of this one, that is polysaccharides. Wish you all the best. Allah Hafiz.